the U.S. Environmental Protection Agency, and they checked the Great Lakes and the history, sediment, the animals.
Awesome. The gallery's so nice.
In the Great Lakes, and it's the largest research vessel that EPA owns and operates nationwide. So the Lake Guardian is designed for long endurance offshore surveys. We can handle a lot of weather. We can be away from port for weeks at a time, and we can bring up scientists. We can get a lot of science done effectively by being offshore for a great chunk of time. So as you can see here, we're 180 feet long, 40 feet wide, with about 5 school buses lengths, over 1,000 tons of ship. We sample all 5 Great Lakes routinely. We're the only research vessel, the only program that does that in all 5 Great Lakes regularly. We do a spring water quality survey every April. We do a summer water quality survey every August, and in between, we spend time in the different Great Lakes depending on our mission. And this year's mission is to do a lot of in-depth survey work in Lake Michigan. So we just came off of a survey looking at sediment contaminants. Cal's talking about the devices we used for that, and we'll talk about that when we get out there. We're getting ready to do next month a big mapping effort looking at the spread of those invasive quagga mussels that live on the bottom. We're going to do a food web contaminant survey and a teacher survey. We bring out 15 teachers from around the Great Lakes to come out and work side by side with scientists so they can take some of this knowledge about the Great Lakes and some of the sampling approaches used out here and work with them with their own students in their own backyard. So we love that survey. So yeah, so we collect vegetable organisms. We've got 3 laboratories that we're going to walk through. We'll be pointing out along the way our rosette sampler, our zooplankton nets that collect the the zooplankton, and then our sediment grab samplers. So a couple of things we have in this lab. This is usually full of scientists. We've got normal tables set up when it's not on a tour. There'll be tons of boxes full of sampling bottles. Fish like to eat them because they're full of fat, so they're very, very nutritious for fish. And they live in the cold, deep bottom waters. But we want to get an estimate of their abundance because fish, it's important to the fisheries. And at night, they come up to feed. They come up to feed on that surface plankton. They come up at night because predators can't see them. We would not be able to catch them. So we have to slow down our speed. We use all red lights on the back deck. We close up all these curtains, and we use red light, which doesn't scare them. They don't see that. If we be caught in here, they don't really migrate up and down the water column so much. So we deploy this in the top 20 meters of the water column. You can see in here, we've got this really fancy looking, looks like a spinner. This is a Swiss-made flow meter. It's kind of like the same precision that might go into one of those fancy watches because this helps us calibrate the amount of flow that goes through the net. So we take that flow rate and convert that with the number of organisms we find to come up with a biovolume. So we count the number of rotations, it logs it, so we know the actual distance that this net traveled. And this is what's going to capture all of our plankton. It gets caught in the bottom of this net, we rinse it down, and we can pour that into a sample jar. And I'm going to show you one of these samples in the microscope in the lab, what that looks like. You see about native species like clams, native worms, native amphipods. Now it's almost like a monoculture in some areas of these mussels. So these are quagga mussels we collected on Wednesday, and they're pretty much everywhere on the bottom in Lake Michigan, especially once you get out past 30 meters of depth. So they're very, very populous right here further. So we spent 2 weeks in Lake Ontario supporting a sonar mapping and video collection effort of ancient shipwrecks on the bottom of Lake Ontario. And here's just a picture of one of them here. There's a lot of ships down there from the 1800s, early 1900s. They're totally covered in mussels, but the cold, deep water really preserves these wrecks well. Warm surface water, that's a great place for plankton to grow, which means it's a great place for fish to eat. So if you've ever gone out fishing and your boat captain's trying to find where the temperature changes, they're probably looking for one of these fronts because there's a lot of biological activity. That lab, the chemistry lab, and the biology lab. And we even bring out a contract chemist to help us run samples. So I mentioned that most of our nutrient samples, we're gonna run back when we get home in a lab on land. It's a little bit more efficient to do it that way. We don't have to generate as much chemical waste on board that we'd have to offload. But there's one nutrient species that we want to analyze out here because it doesn't have a long shelf life, and that's dissolved reactive phosphorus. This is our biology laboratory, and kind of like with a lot of our nutrients, many of the specimens that we collect in our nets or in our phytoplankton samples or in our mud samples, those are going to be most effectively processed back in a lab. It would be really hard to spend all day here. What the compound here is chlorophyll. So I showed you on that screen that green spike in chlorophyll. That's a measure of fluorescence. It's an indicator of biological activity by the plankton. But we're going to actually measure the true chlorophyll units that are in the water. So we bring those samples in here and we can filter the samples and then extract the chlorophyll using an instrument called a spectrophotometer. We do that on board. But if we brought the samples in here and did it in a lab like this with all this bright light, it would influence our results because the plankton would they're still alive and they would keep fluorescing. They would keep making their chlorophyll. And so the measurement of chlorophyll you might get after having your sample in here for a couple hours wouldn't be the same amount of chlorophyll from what we collected in the lake. And we're really trying to understand what's in the lake. So
meals, or if you can look over there before we head upstairs, there's a TV and a lounge area where we have movies playing all the time. I know one of my first surveys, they had a Twilight marathon.
Well, I don't know how many.
We're in the helm now.
Down the sides there a little bit.
Can you hear me?
This is the captain.
If there's shorter people, have them come up to the front, and that way everybody kind of. Oh, my name's George. I am the captain. Downstairs, they showed you the science and the equipment they might use to get the samples and things like that. To get them, they showed you the like an example of the route that we go. They got a bunch of dots all through the lake chart and then zigzag lines for us to connect to them and work our way up and down each lake. So. Is everybody familiar? Those
Drop it right down the water column all the way to the bottom of the lake, wherever we are. And our deepest station is in Superior. I believe that's about 300 meters.
Goes
so
over
go
the
down
side.
pretty far. And then what we're able to do is we're able to bring it up, and we're able to decipher or control when these close at different depths. So number one, the bottle number one over here, is probably going to close around the bottom of the lake. And then as you move up, the number is going to be higher in the water column. So number 12 will probably be ready to surface. That's basically how we're able to all this looks like. I can show you on our way out. But basically, this is where we do a lot of our sediment sampling and our net tows. I know you guys have talked about the nets inside the wet lab briefly. But again, this is stuff to kind of study what's going on at the bottom of the lake. This is the multi-core. So you see, this is an example of a tube in the multi-core. What this thing will do, it'll go attached to this A-frame, get swung out, drop down. And this can actually go into the bottom of the lake and bring up sediment up to like here. So then what scientists are able to do is to take that sediment and then use different methods to date back the sediment back hundreds of years and see what conditions in the lake were like at that time. So this is pretty cool. It can have up to 4 tubes at a time. I like to call it like a spider. Kind of looks a little spidery, I guess. This is the frame for our drop-down camera. So a lot of what we're doing is also sediment analysis, but we're also looking at what organisms are on the bottom of the lake. So we'll attach a camera on the front or on the bottom and on the side. And we'll drop it down to get video analysis of different things like mussels, different crustaceans and worms living on the bottom of the lake. We have a whole benthic group that comes out here. We have a big benthic survey that we give out this year's on Lake Michigan. So they'll do a lot of these deployments as well there. And then this is the box core. Pretty similar to the multi-core, but it's just one big chamber like this that we drop down and we can get sediment about up to here. And it's also pretty useful to see the interface between the bottom water and then all that sediment. So it's a good thing for collecting organisms, collecting surface or bottom water, things like that.
You get to keep it.
Get different organisms. Is this ponar grab? I like to think of it as a big claw. So if you think of like a claw in an arcade, yeah, you just deploy it while open and then we're able to shut it while it's down and it brings all that surface mud and those organisms that were there at the bottom of the lake right back up to us, and then we're able to open it and see the contents in here. This is also where our nets will get deployed as well. So we're able to deploy nets off this A-frame and drop them down and do vertical tows. Any questions about these things before I let you guys go? Yeah.
Anything you do, is it related specifically to climate change or not really?
Not really. We're doing long-term monitoring of the Great Lakes over the past